merge in identifying these and then trying to correlate them with survival data, I think has given a lot of even more interest in this field. So how are these extra chromosomal DNA generated? Well, I mentioned the breakage fusion bridge cycle, but you could have a chromothripsis event, right? These are what I mentioned earlier, where there's a shattering and rejoining of these fragments to generate these complex rearrangements that can generate these circular fragments. And then there are other kind of simpler double-strand breaks that end up fusing into a circle form. And so this is what we see, but how do they really happen? And so I like to just tell you how we looked at this complex patterns of rearrangement and figured out one pathway through which this occurs. So this is just looking at breast cancers and looking at frequent regions of frequent amplifications. And so here, the light green indicates regions with somewhat low amplitude chromosomal gains, and then the peaks, okay, on chromosome 11 and 17 and so on correspond to these focal amplifications. And they're quite frequent. We're talking about 10, 15% of some of these cases. Now, what we really got us going here is that when we look at in the whole genome data, where these are, what we noticed is that these amplifications are actually correlated or connected, okay? So the plot on the bottom, when you see these darker dots, it means that there is a read connecting different amplifications together, okay? So I've always thought of these especially when you were used to looking at array CGH data or cytogenetics data, you think of these as independent events. But when we look at these discordant reads in whole genome data, we see that they're connected, okay? And in particular, you see chromosomes 8 and 11, 8 and 17, 11, 17, those seem to be somehow related. And so that gave us a clue as to how they may arise, okay? The fact that the amplifications are connected at their boundaries. Now, these amplifications are really amazing in some sense because they are recurrent in different cases. So here's one example of a patient with amplifications, right? Those are the peaks at the end of this translocation, containing ERB2 and CCND1. But in two other cases here, exactly the same spot, almost exactly the same spot, have those amplifications and translocations, okay? So what is driving this reproducible event? Now, if you look at the details of the reads connecting these amplifications, it gives some clue to how they may have arisen. So here, I'm looking at one case, okay? You see the orange bars, those are the regions amplifications. You see those tiny little red dots along the amplified region. Those are actually little segments that's amplified. And you can see on the y-axis that the copy number is pretty high, right? So we're talking about 20, 30 copies of that region, okay? Now, these lines that go across, those are correlating or linking, okay, those two boundaries of these amplified regions, okay? And then the red row and blue line at the bottom indicate the major and allele copy number, okay? And so you see that in between the two chromosomes, so you see at the bottom that there's a chromosome 11 and chromosome 6, the regions toward the telomeres of those two, there's a LOH, loss of heterozygosity, indicated by the blue line, okay? Now, the fact that there are so many reads, okay, so on the right axis, you can see the number of reads. So many reads support these translocations. It means that it was actually an early event, okay? The translocation happened, and somehow it got amplified. Not that the amplifications happened, and then somehow they were connected by subsequent events, okay? So that gave us a little bit about, some clue about the sequence of these events. And then the LOH at the bottom told us quite a bit. If this had been a BFB cycle, that some of you are familiar with, this is not the LOH pattern that you'd expect. Now, this is actually the simplest case. I'll show you some other cases. It's a little bit more complicated, and it does actually take a lot of time, months of um, studying these patterns to figure out what's going on. Okay. In this case, again, you see these high amplitude focal amplifications connected by these segments. And without going through too many details here, you can actually reconstruct the sequence of events. It looks like there were 2 chromosomes involved, and there were multiple DNA breaks, okay? Double-strand breaks here in the lines with dotted lines. And then the initial genomic arrangements happened as shown. Okay? This is all inferred from the patterns of the discordant reads. And then it looks like there was a dicentric chromosome formation. Okay? So you have one dicentric, then one acentric chromosome that usually gets lost. And then in mitosis, they will get pulled apart, and then just like in BFB, there will be lots of breaks. Okay? And then the fragments that get broken off, they will often go on to form ecDNAs. Okay? So I've been actually staring at these really complicated patterns for a long time, but until this postdoc came along and really spent a lot of time looking at this, I did not think it was possible to tell what's going on. But once we figured out what the sequence may have happened, and I'll tell you the model in a minute, we think that it actually goes very well with what we see in this complex pattern. Now, that was the case that I just showed. Sometimes you can have multiple chromosomes. So here there are 4 chromosomes that are involved. There are lots of pieces that get fused, but the common thing is that it's formed. One of those end up being a dicentric chromosomes that again go through this breakage. So what we find is that unlike the breakage fusion bridge cycle, okay, there's something else going on that generates that specific pattern. So just to, again, remind you, the way that BFB cycle happens is that you have a double strand break, okay, and then it gets replicated before the breaks are fixed, right? The timing of when the double strand break is repaired is really important to what the final form looks like. So there's a replication here, and then there's a fusion, and then that forms a chromosome bridge. In our model, which we call translocation bridge model, because the initial steps involve translocations, there are actually 2 chromosomes. There are multiple double-strand breaks. Okay? And then they get fused. They don't always have to fuse this way, but sometimes they'll get fused in such a way that there's a dicentric chromosome. Okay? And then there's replication. Okay? And then in some of those cases, the microtubules will attach to kinetochores in such a way that when they are attached to the opposite poles, it gets pulled apart. Okay, so we call it anti-parallel direction. Okay, so it's similar to the BFB cycle, but here there are 2 chromosomes, and then they get pulled apart. Okay, and then we get the pattern that we see. Okay, so it's similar in some ways, but again the point of when double-strand breaks get repaired makes the difference. Now, the question was then, well, why did these double-strand breaks occur in the first place? Okay. So we tried to get all the epigenetics data available and then try to correlate them with the sites of the initial break. And it turns out that estradiol-treated cells have ER-alpha binding sites that correlate really, really well. Okay, and so this is some multivariable model with p-values. So overall, across all the tumors, all the breast cancers, there's high correlation with this ER-alpha binding site. But ER positive cases actually have very, very high significant association, whereas ER negative cases have basically no correlation. So there's some correlation with ER alpha binding that's really obvious, right? And then you have CTCF binding sites, then you have topoisomerase 2B, and so on. So these are the factors that appear to be associated with the initial site where double-strand breaks occur. And we can actually look at the ER alpha binding sites, okay? And this actually surprised me because we typically think of, when we look at ChIP-seq profiles, lots of sites that are bound with, equal strength. But here, you see that the ER alpha binding intensity is really, really high for a small set of sites, okay? And so for instance, this RARA gene is right next to ORB2, okay? So somehow there's something going on, open chromatin, certainly, but somehow there's a gigantic fraction of cells in which ER alpha is bound, okay? Now, and so that to us, that it provides some explanation for why these amplifications occur at specific sites across many, many tumors. Now, of course, we submitted this paper, and one of the reviewers said, "Okay, can you do some experiments?" Okay, so this was the pandemic era, and so it took some time. But luckily, we worked with Roberto Chiela's lab at Boston Children's, where they had developed, when Roberto was in Fred Zelt's lab, developed this assay called high-throughput genome-wide translocation sequencing. It's basically a way to enrich for translocations, okay? Because you don't want to sequence the whole genome and then find a few, right? You want to enrich for those translocated fragments. And so what we did here was treat some breast cancer cell lines with estradiol, and then generate a CRISPR breaks at sites that I showed on the previous plot, next to ERB2, next to CCND1, where we see high binding. And then we try to see where those fragments will fuse, okay? So there are two experiments that are shown here. We generate the breaks at the gene RARA and SHANK2. SHANK2 is next to CCND1. And then it turns out that when you generate those CRISPR breaks, indeed, they tend to fuse with specific locations in the genome, especially with the partners that we see in real tumor data. And when we do the same kind of analysis, again, yes, when the sites with CRISPR breaks tend to fuse with sites that are bound by ER alpha, and so on, okay? Now, there's actually a lot of biology here that I'm skipping. So there have been literature going back 15, 20 years about how ER alpha binding generates double-strand breaks in nearby regions. And there's a paper from a lab here at UCSD, Rosenfeld lab. There have been some other papers on how you need these double-strand breaks to relieve a sort of topological stress in transcription. And so it's not totally new that somehow ER alpha binding causes double-strand breaks. But this is sort of one result, one consequence. The patterns of translocations we see is one consequence of that breaks. Now, we did go on to look at lots of other tumor types to see whether this pattern of translocation bridge amplification can be seen seen across tumor types. And so here we looked at 2,000 plus genomes available in ICGC. And so here I'm listing all the tumor types on the by rows, and then chromosomes along the x-axis. And then we could, in fact, cluster different tumor types into the types of amplifications observed. So in the top, sort of marked with a red bar, those are the tumor types in which breakage fusion bridge cycle is very common. Okay, so the way to detect it is this fold back inversion patterns that people know about. And then there's kind of mixed group where lots of different things happen. What I've been talking about with breast cancers, but it also occurs in liposarcomas, bone cancers, and so on. These are the group of cancers where this translocation bridge amplification is very common. Again, why is that? What's so different about those tumor types? On the left, right, we classify them into the BFB cases where the repair happens in SRG2. In the TB amplification cases, the repair happens in G1. Okay, and somehow these tissues have different timing of the repair. Now, you could still ask why those sites in particular. Okay, so I mentioned the very, very strong ER-alpha binding, but it certainly also has to do with tissue type specific chromatin features. So for instance, here we looked at DNA's open chromatin mark in these two, I'm showing just one cell line data, T47D. Without estradiol treatment, there's no open chromatin at those sites that we typically see the translocation. As soon as we treat them with E2, we see that the chromatin opens up. And when we look at the binding of ER-alpha, there's no binding before treatment. And as soon as the chromatin opens up, it seems to be that there's a lot of binding. Okay? And so, so we still have a lot to learn. You know, why? Because one of the most common questions, well, are there other factors other than estrogen, right? Other hormones, right? In other tumor types, what's going on there? You know, why don't we see the same patterns? So there are lots of questions that we can discuss. But just to summarize, we do think that this is really frequent. Like in the breast cancers, the part of from the pie chart, about a third of these cases, right? But about 2/3 of breast cancer cases have these focal amplifications. About half of the focal amplifications arise from translocation bridge amplifications. That's a lot, right? A third of these cases. Complex translocations, chromatin bridge, focal amplifications, and then it's really correlated with ER-alpha binding. And then we can see that easily partly because we see that ER-positive breast cancer cell lines are where you see this, and then there's a tissue-specific mechanism. Okay, so all this work was led by Jake Lee, a phenomenal physician scientist. A lot of help from David Pellman, Steve Allergy, and Robert Giale. And this is one of these cases where we got a lot of reactions from people. There are some clinicians who called me up and said, well, we do estrogen therapy for some breast cancer cases. Does this mean that we should not treat them with estrogen because they will generate additional DNA breaks? I don't know the answer. I think there's a lot of clinical trial data on this with mixed result. So I think there's a lot to be learned about the whole process. Okay, yeah.
I'm curious, do you see patterns with like fidelity of repair pathways, like associated with like the patterns of tissue of complex fragments in the complex types?
Yeah, that's a good question. So we have tried to correlate with lots of other annotations of those tumor types or tumor cases. And so, for instance, in HR deficient cases, we don't see this so often. So HR has to be really active for the repair to happen this way. Other than that, there aren't too many other, repair pathway deficiencies that we could correlate with this. Yeah.
So did you already like this as a way to diagnostic
Well, I think for PARP inhibitors, that's actually another topic that I could spend the whole seminar talking about. There are some other ways to predict PARP inhibitor efficiency, and so I'm not sure how this would really fit in. But yeah, I think for that, there's a lot more direct way of looking for HRD cases and so on. Yeah, it's actually a very complicated topic because even among the HRD cases, who are usually considered for PARP inhibitor therapy, Some respond and some do not. So we do understand that there's a lot of heterogeneity, lots of factors that we haven't looked at. Yeah. I'm going to talk about brain-related things next. So if there are questions on this topic, maybe I'll take a couple more questions now. Yeah. Well, I suppose that could be related to how much amplification you see, what fraction, right? What fractions of cells have that? But the initial steps, right, is what we're trying to describe here. Yeah. Okay. Well, you can still ask questions about this topic at the end. So for this part, I like to talk about somatic mosaicism in the brain. So Joe Gleason's here. I had a chance to work with him a little bit through a consortium. There may be other experts here, but I'll try to give you my take on this. And so, just like many other things that we do in the lab, I got into this because we had developed some tools for cancer genomics, and then the people working on the brain side came to me and said, "Well, could we apply that technology for the brain data?" Somehow, there's a bigger community perhaps. There's a lot of technology development, some of which have filtered down to other areas. But this was, I think, a really an exciting area that more cancer, methods developers should get into. Partly because the problem's harder, right? Because the mutations will often not proliferate. The cells carrying the mutation will not proliferate as in cancer, and so they're rarer and harder to detect. We got into this looking at transposons, but the problem itself is more general, right? These post-zygotic mutations generate lots of different distinct populations of cells, okay? And as I'm coming to learn, using these somatic mutations as markers can answer a lot of fundamental questions in developmental biology. I wish I had more time to talk about that aspect of it. Okay, so there's just a general interest in the development, what tissues arise from what, cellular lineage and so on. And of course, there's a link to brain-related diseases that got us really interested. Now, there are multiple ways to study somatic mutations in the brain. Okay, this is from a review perspective paper. So one way to do is just throw money at the problem, right? That is bulk tissue sequencing really deep. Okay, and so we have rather than 30x data, 250x data. Okay, so not cheap, right? So that's one way to do it. But you can only sequence so much, right? Even if you sequence 10,000x, you're going to miss a lot of mutations, right? So with 250x, we really do try to come up with more sensitive algorithms, because if you can have an algorithm that can, have 50% greater power, then you're going to save, millions of dollars for the field, right? So we're still working on that. But the really rare mutations you cannot solve by sequencing deeper. So one way to do this is to do clonal expansion. Take single cells, expand them clonally, and then you have enough to do whole genome sequencing using the standard technologies. That's a little hard to do because you can't always expand cells, right? You can't expand neurons too easily to this approach. And so the approach that we've taken is to isolate neurons, amplify them, okay, and then do 30x sequencing, basically, right? So rather than doing a bulk 30x, we do single cells, DNA amplification, and then 30x sequencing. Well, it turns out that this is a really challenging problem bioinformatically, right? Because the noise that gets generated from amplification will overwhelm the true mutations, okay? All the work that we've been doing in the brain space is with Chris Walsh at Boston Children's, and a lot of people from his lab. Now, just to illustrate how finding these really low VAF, variant allele fraction, how these rare mutations is so hard. Um, in this consortium that Joe is part of, this is actually probably maybe 7, 8 years ago now, 5 labs in the consortium were given basically the same sample. Okay? And we said, okay, find all the somatic mutations you can from this. Okay? And they did, some did whole genome, some did exome, some did whatever. Some used some matched normals from what they had in their lab. And they came up with 5 sets of variant calls, and the result is what you see. Okay? All over the place, right? And the funniest thing about this is that one thing that's common was actually a mistake. It's a misalignment due to a germline insertion, okay? So you think calling somatic mutations cancer is difficult, right? This is much harder, right? Because you're really trying to drill down to, maybe you can catch something with a single read, okay? Maybe 2 reads, okay? And so we spent some time working on this. But no matter how good you can get, again, you really need to look at single cell analysis, okay? Now, I'm not going to talk about the details of it. I mean, if there are questions, I'm happy to do that. But initially, we thought, okay, one thing we can do with this technology is to look to see whether mutations at the single cell level, mutational load at the single cell level changes with age, okay? And so we have few people here, 15 individuals, so each column shows different cells from the neurons from the same individual. So there's some variation within an individual. But overall, there seems to be an increase in the number of somatic single nucleotide mutations. And so the estimate there was about 23 somatic mutations per neuron per year, right? And so remember, the neurons are not replicating, right? These are not errors from replication. They're just it turns out they're somewhat related to transcription to a small extent, but these neurons are just accumulating mutations, okay? Now, after that, we came up with, we ran into a technology called PTA. This is now, has been commercialized by a company called Bioscribe. But we worked with them from the very beginning, and it turns out that this PTA is a better amplification method, okay? And so compared to multiple displacement amplification that we used to use, PTA generates a more even coverage, okay? And so what that does is that, as you can see here, bulk sequencing gives you pretty even coverage throughout the genome. Very common method called multiple displacement amplification that we used to use, as many people, has a lot of noise, right? Some regions of the genome are just amplified more often, okay? And then this new technology, PTA, generates a more even coverage, okay? And you can see this on the distribution of the allele fractions here. And so in bulk, you see the variant SNPs at 0.5, and the distribution is not as good as bulk, but PTA gives a much better result than MDA. And so using the MDA data, we try to come up with a way to call mutations, right? The standard GATK or any germline variant calls, if you apply to the amplified single cells, 95% are false positives. Just can't do it, okay? And it has to do with just the fact that some alleles are amplified at a faster rate than the others, and it's hard to tell, okay? And so we came up with a way to call this mutations. We think that it generates 60-fold fewer false positives per megabyte than conventional method. And so this was just so that we can do lots of interesting, variant analysis afterwards, okay? And so one thing that we were able to do was with this PTA amplification technique, we could in fact modify the previous estimates a little bit. And so we think actually the more correct rate is about 17 somatic SNVs per year per neuron. We actually could call indels pretty accurately, and we see that there are some indels as well. Now, one thing that's interesting about this is, we have to, if once we found out not just the mutation load, but where enough mutations are found, we could see whether they have any functional impact, right? Because that's ultimately the question, right? Do these mutations do anything? Well, it turns out that these mutations are enriched in brain-specific promoters and enhancers. Okay, so on the left, you see different cell types here. Um, you see some enrichment for single nucleotide variant here in neurons and oligs, okay? But really, indels are enriched in enhancers, okay? Well, it turns out it's not just any promoters or any enhancers, because we can look at tissue-specific enhancers, and it's only in the brain-specific enhancers these mutations are found, okay? And promoters also. Now, there is some literature on how neural activity can trigger DNA breaks and so on, and that's perhaps the mechanism by which these indels are generated in promoters and enhancers. Um, so, um, so it does make sense, but we are able to see this in single cells. There are so many things that we could do with this technology. I didn't put it in the slide here, but one of our initial work was to sequence brain and other cell types, and then identify these mutations, and then do a panel sequencing of lots and lots of tissues, okay, for those mutations. Then we can actually figure out some lineage information, right? This is sometimes called lineage tracing, right? And so these somatic mutations are markers by which we can tell how uh, in development different lineages were formed. So for instance, we have a paper in bioRxiv where we look at different brain sections. And so there are certain brain regions that do not share a lot of variants, and other regions that seem to share a lot of variants. And so we know that that means that those two are closer in development lineage. And so here's another example of what we can do with the mutations that we identify. And so we look to see whether neurons and oligodendrocytes have different rate of mutation and different locations of those mutations. Okay, and so here again, x-axis age, and you can see that the point mutations increase in both cases, but in neurons, the rate of mutation is lower. So in oligodendrocytes, SNVs accumulate 80% faster, and indels accumulate slower. Okay, now why is that? Maybe relate to transcription, but certainly there are lots of things that we don't understand. Another analysis that we can do is to look at these, the distribution of these mutations. And when we look at oligodendrocyte mutations, we see that they're enriched in inactive genomic regions. Okay? It turns out that this is exactly the pattern that we see in brain cancers. And these gliomas arise from oligodendrocytes, so it makes sense. Whereas the neuronal mutations are in open and transcriptionally active regions. So just by looking at these single cell based point mutations, we can tell a lot about what's going on, the different processes in different cell types. And so there's a lot more. So we do a lot of mutational signature analysis and such. It's all described in the paper. I forgot to put the reference here. Came out in Cell last year. Gans, G-A-N-Z is the first name, or first author. Okay. And so there are lots of other applications of this. I think we still don't understand what fraction of cells should have certain mutations before it becomes as functionally important. But certainly the fact that some of these mutations, some of which are subclonal, some of which are really specific to a cell, the fact that they're enriched in these functional regions tend to think that at least some of them may be functional. Now, just to tell you some of the things that we're doing in the lab now, we are part of this consortium called Somatic Mosaicism Across Human Tissues. I had nothing to do with the name, SMOT. Somehow NIH came up with this. It's one of these common fun projects, ENCODE and GTEx and roadmap epigenomics are all sort of these, common fund projects where different NIH institutes pull money to do a project of shared interest. It's one of these projects. It goes for 5 years, potentially for another 5 years if the first phase is successful. But the idea is to better understand somatic mosaicism across different disease as well as in development and aging. And so I'm talking about this because it's going to generate a lot of interesting data sets that some of you might be interested in analyzing. And so the idea is to sequence lots of tissues. 15 is the ideal goal across lots of individuals. Okay? And we want to throw all the latest technology to profile these tissues. Okay, so Illumina data, 300x whole genome. We have a lot of debate about whether that's a waste of money or if it's deep enough. We also do PacBio whole genome sequencing, Oxford Nanopore. There's a class of methods called duplex sequencing that's becoming very popular. We think that it's going to be a major technology to understand mosaicism. Just to figure out what the best tools are, we have to generate some truth set and test various methods. And so we have some benchmarking data where we have 2000x whole genome, right? And 300x PacBio and et cetera. And so there's a lot of data. This right now is consortium only, but it can be shared very, very soon. And one thing that we are really interested in doing is to do donor-specific genome assemblies, right? That is ultra-long ONT and high-C combined with PacBio and Illumina can give you almost T to T quality genome for individuals, okay? So this is very, very new. I mean, I have a postdoc in the lab who's been testing different methods. There are a couple assemblers, but it's surprisingly good, right? And one major advantage of this is to look at all these repetitive regions that you just could not with the Illumina data, but also even for HG 38 regions, these regions that previously were not part of the assembly will get all these reads, will prevent these reads from misaligning to the common reference genome. So you'll actually get rid of lots of mistakes that we just can't avoid with Illumina data. It's primarily due to misalignment. And the extent to which that this has been a problem, I did not appreciate until we actually mapped this. The other thing about this is that we are generating diploid assemblies, right? And so the fact that we can look at allele-specific regulation, I think, is going to be really important. Okay? And so that's brand new for us. I mean, we don't even know how to align to a diploid genome correctly. I mean, we can do some sort of, we can take some shortcuts and, put the two haplotypes together into one contiguous one and align and such, but that's not optimal. And so we're trying to figure out how to align to the genome. Lots of other techniques being developed. So you can see the assay types here. And then we hope to look at lots of other molecular features. And so all this is coming. And so we're just in year 2. Hopefully, we can make this public very soon. The hardest part actually is figuring out how to do all the paperwork, right? Because these are, human samples, what the latest NIH requirements about. Okay, don't get me started, okay? It's driving me crazy. Okay, but once that's taken care of, we like to share as much data as possible with everybody. Okay, so a lot of people in my lab have contributed to this. I took everybody to Florida at the end of the pandemic. There's a picture. And Chris Walsh, again, is a major collaborator, and as well as lots of people locally and the SMART Consortia. So I'll stop here. So thank you very much.
Yeah, I'm wondering if you could say a few words about the mutational signatures. You sort of said were in the paper. I mean, and a question, do you see a lot of cytosine, guanine mutations? Because the hypothesis would be that cytosine methylation over time is naturally deaminated, and that's the source of a lot of somatic mutations, at least in cancer genomes. So I'm just curious if you see a similar effect.
Yeah, so we see signature 1 and 5.
Yeah, that's signature 1, basically.
Yeah, dominant also in neurons and oligodendrocytes. There are some other signatures that are a little different. SBS 19 and I think 32 and so on. So we're trying to understand why they're a little different. But but the majority are
So,
CAT.
so look, it'd be really interesting in this next project if you added DNA methylation to the list of omics. I didn't know if it was a long list. I didn't, it might've been at the end there. I don't know.
Yeah, so FiberSeq is actually,
Okay, great.
yeah, it will give you methylation data, and there's something else that will give you different flavors of methylation too. Yeah.
Hello, wonderful talk. I was just wondering if when you're looking at the different types of mutational burdens between the cell types, if you're also looking at mutations that might be in the UTR regions of these different cells. And if you're also planning on using any of the long-read sequencing data to look specifically at the different RNA isoforms between cell types as well too.
All great questions. So, so we can do pretty good job with point mutations. The reason that we actually get these more even coverage with this new amplification method is that you force the fragment to stop amplification. So it becomes kind of a semi-linear. And so what you don't get is longer fragments that will allow you to do SV analysis, right? So that's a trade-off. We would love to be able to generate long fragments that we can do PacBio on. Okay, that will solve lots of problems, but it turns out that it's not that easy to do. In terms of RNA, yeah, I'm totally, into generating PacBio RNA data because all the things relate to splicing that we used to do with short reads. I mean, we could do some guesswork, right? So much cleaner with, PacBio long read data. And so we are, trying hard to do that. And it's actually so the PacBio Kinnex kit was, became available early last year. So it's all new. Before that, it was, too expensive, right? Basically, there's some protocol where you concatenate lots of different fragments into a single one that you can put into PacBio that will lower the cost. And so, yeah, so I'm sure there are lots of labs that are really trying to do this. So we have some point mutations that we can find, and UTRs, yeah, we can certainly look at them. I don't think we have enough to do lots of significance analysis yet, but I think as soon as we have more cells, we think we can do that. And then the fragments, if we can sequence longer fragments, it turns out, and I'm actually learning this, for this SMART project, generating long read whole genome data is very difficult for some tissues. They just cannot get these long fragments from certain tissues. And so they will, for cell lines, you want to get 30x PacBio? Yeah, just put it on the machine. For some of these tissues, you try to do 30x, they'll give you 1x. They don't know why. They just can't get the fragments to do PacBio sequencing on. So it's much harder. And I'm not talking about in the small labs here. We're talking about, the WashU and Broad and New York Genome Center. They're all having the same problem with tissues, okay? So short read, no problem. Long read, I don't know how much data we're gonna get. So that's like, we're arguing on the call, like, every other week on this. Okay, so to be determined, yeah.
Thank you very much. See, Paul Love's hand was up over there.
Thank you. So it seems like the sort of underlying mechanism for generating these breaks, both in the estrogen-mediated pathway and also in the promoter-specific breaks that you identify in the neurons, it seems in some level related to topoisomerase. Is that correct? So both estrogen recruits topoisomerase, and if it's you have a lot of transcription that is initiated at those promoters, you need topoisomerase to relieve the stress. Do you think topoisomerase could be the sort of common culprit for generating these breaks?
Well, we certainly, that was like number 3 on our list, right? Right. So, you know, I would love to know a little bit more about exactly, you know, how that interplays with other factors we see. But certainly it appears to be the case.
So it's probably topoisomerase plus something else that makes it fail, right? Because normally it does this all the time and it doesn't break.
Yeah. So, yeah. Maybe some of you know more about this than I do. But yeah, totally. I think that's um, yeah, that's an area of interest. And yeah, so it's something that we would like to pursue, but just because we don't have a lot of expertise in the experimental aspect of this, I'm not sure how to best go about doing that.
Thank you. Maybe a bit of a related question. So I know it's been shown that in some early replicating fragile sites, these occur because there is a physical collision between transcription and replication in highly transcribed regions. So I was wondering if, in particular, for the first set, or the first story you showed, if these places where the focal amplifications are occurring more frequently, are they correlated with being nearby highly transcribed genes or estradiol upregulated genes?
Well, so those sites are more frequently found in earlier replicating regions, but I think the exact sequence of events, it's a little bit unclear, right? Because we're seeing correlations, right? Is it the DNA that opens up, right? That then allows transcription and the factor to bind? Could there be some other sequence of events that can drive this? I'm not sure. I mean, we can guess. And so, I don't know whether it would take certain experiments to show the sequence of events, but yeah, they're all sort of correlated, right? You got to have an open chromatin, you got to have the factor bound and early replicating regions, the topoisomerase, transcription. The we did look at transcription level of these. I mean, usually like these amplified oncogenes, they really have high expression levels, right? And so that's clear, but besides that, I'm not sure what else to correlate. Yeah.
Maybe time for one more question. Well, I had a question. You mentioned that these misalignment artifacts are a big problem in somatic mutation callers. The fact that certain reads might misalign to the reference genome.
Yeah.
So are you suggesting that even with the matched normal, the misalignment will only occur for some reason in the, non-normal, kind of experimental tissue?
Yeah. So, great point. So if you have a matched normal, that will get rid of most of the misalignments, right? Because whatever misalignment you get in your sample, you should find that reproduced in the matched normal, okay?
Yeah.
But that's in tumors, right? Where you could get matched normal.
I see.
In the brain, what's the matched normal, right? So that's the problem. That's the
Okay.
problem that we have. And of course, you can get lots of different tissues
Right.
and then identify tissue-specific mutations by using the others as controls.
Yeah.
But if it's in early developmental mutations, you don't have a matched control. So that, I'm not sure how to solve that, right? Yeah. And so, one reason that PacBio is really nice is that you can phase these mutations, right? And so we're actually finding that so many, so with the data that I mentioned, what we've been doing is looking at all the mutations generated by Illumina, looking at all the mutations generated by somatic mutations generated by PacBio, and trying to figure out what the difference is and why that happens. It's certainly true that so many Illumina-based mutations go away when we align the reads better with PacBio reads. But even some of those common ones could be mistakes because we can look at other data or phasing, okay? But certainly the phasing, we used to do phasing in single cell data with short reads, and we could only phase about 15% of the genome. But with PacBio reads, we can phase most And so it's so much better. And so that's another reason to the PacBio if you are trying to really increase specificity of your mutations. Yeah, good point.
Great, thanks so much. Let's give you a
Thank
round of
you.
applause.